Our bodies are equipped with an incredibly smart, multi-step mechanism of fighting disease and protecting against them. Our bodies are able to do so due to our immune systems that allow us to come in contact with billions of pathogens a day and still not get sick. It does this with its three lines of defense. The first is your skin, hair, sweat, and mucus that act as barriers to keep out pathogens and trap them in an area to be expelled. The second is your nonspecific barriers, which use phagocytic cells like white blood cells and other macrophages to identify, engulf, and destroy a pathogen to prevent infection. Say you get a paper cut. A pathogen is able to break past the first barrier and enter your body. Mast cells sense the intruder and send out histamine signals to attract the macrophages and to the area. The phagocytic cells identify the pathogen and engulf it, digesting the pathogen. Now from here, it meets the third line. This one is specific, meaning it doesn't just attack anything. The cells have specific receptors to bind to specific pathogens to defeat only those. When a pathogen infects a cell, it is able to break down and send out a sort of signal to alert other cells. This is the MHC molecule, which is attached to a piece of the broken down pathogen known as the antigen. A helper T cell comes along and binds to the MHC molecule, which causes the cell to release cytokines and activate the helper T cell. The antigen-presenting cell leaves and eventually dies, while the helper T cell begins signaling clonal selection of itself or activates a B or cytotoxic T cell. The active cytotoxic T cell finds an infected body cell and binds it to MHC2 molecule. It releases cytokines and enzymes called perforin to poke holes into the cell and cause apoptosis to destroy the infection. However, when a B cell is activated by a helper T cell, it begins by searching the blood for any pathogens. They are covered in thousands of different antibodies meant for a specific pathogen, and once it finds its match, it binds and signals for the helper T cell once again. It then binds and releases cytokines that signal the B cell to begin cloning itself. The clones form, but then differentiate into two types, memory and plasma. Memory cells are covered in the antibodies for the pathogen and stay in the blood to catch the same pathogen if it enters again. The plasma cells begin producing free-floating antibodies that also circulate the blood to locate and surround any pathogens to signal to other macrophages that they have something for it to destroy. The cells and antibodies split up and go on the hunt for more pathogens.